Hi. So I saw that Dr. Layne Norton released a video on him being very skeptical on collagen supplementation based off of a brand new meta-analysis that released. And it turns out that I have access to that meta-analysis. And I thought that I would react to his claims related to collagen supplementation and see if we agree or disagree. For the record, before we start, based off of my analyses looking at, this is specifically for collagen on skin, my interpretation of the literature has been leaning in the direction that collagen, specifically collagen peptides, are effective for reversing skin aging and wrinkling and improving hydration and things of that nature. So we'll see if that still holds true. And we'll also see what points Dr. Layne Norton has. I think in the past he's been pretty skeptical in general, but now maybe he's even more skeptical based off of this analysis. So let's get into it. And I'm actually going to be releasing a more substantive video on this very topic for the main channel in the near future, looking at his video. Okay, let's get into it.
Collagen supplementation looks like it's a scam. One thing I've been very consistent with is I am very skeptical of collagen supplementation for the following reason. You trying to improve your connective tissue or your collagen. If you eat collagen, if you ingest a supplement, that supplement gets broken down during digestion to the constitutive amino acids. It's not like you take in collagen and it just goes right into your connective tissue.
Okay, so let me pause there for a second because he's somewhat correct on that. And I'm a little surprised that he's making the argument that way simply because I've heard some of his other content on collagen supplementation where he was still skeptical, but he did acknowledge that di and tripeptides can enter into our body. So what does that mean? So you've got a collagen protein, which is this lengthy protein. It's made of all these fibers and whatnot. And so it's got hundreds of amino acids. So that's the constitutive, as he said, the actual base pieces that make up a total protein. And it makes sense that we wouldn't be able to absorb. So from, you consume it, you mash it up in your mouth, it goes into your stomach will denature it, but it'll still be bound in this kind of polypeptide chain of many amino acids. And then it'll go into your intestines, and there it'll go through further processing as he described, and then it'll be absorbed. And usually most proteins are absorbed as single amino acids. So all those amino acids that were linked together get broken up into single amino acids and then they get absorbed. However, that's not entirely the full story anymore. And we know that from human research as well as animal research that it is possible for our intestinal cells to take up amino acids, not just as single amino acids, but also as di and tripeptides. And what that means is that you have 2 or 3 amino acids that are still chemically bound to one another. So they're bonded together. And in addition to that, there's some idea that we can get even larger peptides that also go through our intestinal tract. Not necessarily full-on proteins, but a little bit more than just the single amino acids that Layne is talking about. And in this picture is a great example of what I'm talking about. This is from a review. And what they're showing here is that, yes, you can get single amino acids. So here we've got the intestinal side so where all the food is and it's being broken down. And then it's trying to get across, just follow this black arrow to the inside, to the actual bloodstream. And then it goes through the bloodstream and interacts with all the different cells of our body. So it's true that single amino acids get through, and that's what we've known for a very long time. But on top of that, we do have peptide transporters, which allow for these di, again, 2 amino acids that are bonded together, and tri, so 3 amino acids bonded together to get through. So we've got specific transporters that allow that to happen. We also have in the small intestine, because your epithelial cells that make up your small intestine, so these are the cells that line your intestine, they are bonded to one another. So they're kind of hooked onto one another. And the way that they're hooked onto one another is through these proteins. One of those proteins is called a tight junction. So they can actually loosen the tight junctions or tighten the tight junctions. And typically with a healthy epithelium, you typically have tighter junctions. But that doesn't mean that there can't be exceptions where you have a looser tight junction and you have a little bit more space there that might allow some of these larger peptides to get through as well. But even if we were to remove that aspect out of the equation and just focus on the peptide transporters, we do have a known mechanism, a known way by which peptide transporters will allow peptides, 2 and 3 amino acids bonded together, into the bloodstream. So I don't agree with this characterization that all proteins or even the majority of proteins necessarily get cut down to just their single amino acids, and because it kind of creates this false foundation for an argument. I wouldn't call it a straw man because there's a lot of truth to it, but there's some aspects there that are being looked over.
That's not how it works. It gets broken down mostly into single amino acids. Now, can those single amino acids be incorporated into collagenous tissues? Yes. But just because you eat more of it doesn't mean that you will increase the rate of connective tissue or collagen synthesis.
Okay, so he's absolutely right about that. It's true that if we were just to focus on the single amino acids, that just because you consume single amino acids doesn't necessarily mean that that's going to stimulate synthesis of the actual protein that you want. Remember, if you consume collagen in its full form, full protein form, it goes through all these modifications that ultimately break it down to, in the best case scenario, let's say di and tripeptides. And then those get absorbed and then they go into our bloodstream and then they can interact with our cells. And I think what Dr. Norton is talking about is that the cells would then take those single amino acids, maybe they would further break them down, or in the worst case scenario, what he's describing is we only have single amino acids, that those would then get taken up by the cells, and then they have to actually synthesize a whole nother collagen protein. So it has to go through this whole breaking down process and then be reconstructed on the inside of our body. So it makes complete sense, and I completely agree with them that just because you supply the amino acids doesn't necessarily mean that you're going to get increases in synthesis. That there are exceptions to that with certain amino acids like leucine, which collagen does not contain in any appreciable amounts. So it's kind of a moot point, but you have exceptions to that, like leucine, which is a highly protein synthesis stimulating amino acid, which ultimately does lead to more protein synthesis. But that's not what we're talking about. We're talking about collagen, and those amino acids don't necessarily guarantee that you're going to get more collagen synthesis as he's explaining it. Again, that's leaving out the potential for the di and tripeptides to have an effect.
Because we do have human randomized controlled trials demonstrating that collagen supplementation can improve pain, recovery outcomes, that it can improve skin elasticity, skin hydration, reduce the risk of wrinkles. Why is there a divergence in this research literature? Well, a new meta-analysis just got published looking at the effect of collagen supplementation on skin elasticity, skin hydration, and wrinkles. And the overall take-home from this meta-analysis was that collagen supplementation improves skin elasticity, skin hydration, and decreases the incidence of wrinkles. So that means I'm wrong about collagen supplementation, right? Hold up, hey. Just reading from the discussion of the paper. However, in the sensitivity analysis, now a sensitivity analysis is where they remove each individual study.
There we go. Done. End of the video. We can go home. No, of course not.
Hold up, hey. Just reading from the discussion of the paper. However, in the sensitivity analysis, now a sensitivity analysis is where they remove each individual study one at a time and see if they redo the meta-analysis if the results still hold. And it's meant to protect against a single study having too much influence on the overall meta-analysis. So Again, I read, in the sensitivity analysis, excluding outlier studies showing an extreme beneficial effect, collagen supplements showed no significant improvement of wrinkles and a decreased effect for skin hydration and elasticity. So basically, if you look through this meta-analysis, there was one study that showed like this huge benefit. And when they took that out, it either nullified the results or drastically decreased them.
Okay, so he's absolutely right about that. He actually explains it really well, that when you're doing a sensitivity analysis, you're removing single studies to see how much impact those single studies have on an overall analysis. There's more to it than that, like the weighting of a particular study. If a particular study is way out, it shows a massive effect of collagen, which to be clear, that did happen in this analysis, that they did have a few studies that were way out there. And when they removed them, to Dr. Norton's point, there was a lessening of the overall effect that they were able to find. But like I said, there's other factors that go into that that are beyond the scope of this discussion. But in general, he's absolutely right. Sensitivity analysis showed a decrease in the effect. And I think in one situation, it led to a nullifying of the beneficial effects of collagen.
But more importantly, they did what's called a subgroup analysis, where they add an extra differentiator in the inclusion criteria, and they separate out these different studies and look to see if the results still hold. Their differentiator was funding source. So they separated out studies funded by companies that sell collagen skin products versus studies that were not funded by companies that sell collagen skin products. And what did they find? I read direct from the paper. More importantly, in the subgroup meta-analysis by funding source, studies that did not receive funding from pharmaceutical companies showed no significant effect of collagen supplements for improving skin hydration, elasticity, and wrinkles, while those funded showed significant improvements in all categories.
Let me pause right there. I just want you to keep the word significant in your mind because it's going to be important in my next point.
Then they did one more subgroup analysis. They did it based on methodological quality. I will read again from the conclusion. High quality studies revealed no significant effect of collagen supplements for improving skin hydration, elasticity, and wrinkles, while low-quality studies did show their beneficial effects for improving skin elasticity. I do say all the time, hey guys, if your only criticism of study is the funding source, it says more about your bias than the researchers. And I stand by that if your only criticism is of that. But those studies also, in this meta-analysis, were of lower methodological quality. So whether it was studies were not randomized appropriately, the individuals were not blinded to the treatments, or the researchers were not blinded to the treatments, those can all increase the risk of bias and lower the quality of the research. And the point is, replication is the mother of all science. If an effect is real, it will be consistent across subgroup analysis and sensitivity analysis.
Okay, let me stop there. So he's talking about a subgroup analysis, which he, again, explained it quite well, but in case you missed it, basically they're taking the full pool of studies that they have, and then they just segment them based off of, okay, in this bucket, we're going to put all the industry-funded studies, and in this bucket, we're going to put only the studies that were devoid of industry funding. And that's a great subgroup analysis. And to his point, the researchers did write what they wrote. They said that there was no statistically significant effect found when looking at the studies that were specifically in industry or in non-industry funded studies. So that would then, to his point, which I completely agree, I don't discount, or maybe I'll discount, but I won't completely eliminate studies simply because of their funding source. And I do think that people that are biased will usually hide behind that as an excuse as opposed to critically analyzing the study. However, let me show you something from this study that he doesn't mention. Okay, so what we're looking at here is the characteristics of the included studies. And here you can see funding from pharmaceutical companies right up here. So anytime you see no, that means that it was not funded by industry. And here we've got the main findings. I'm going to bear with me because I'm going to walk through each one of the studies that was not funded by industry. So this one, this Sugihara et al. Study, was not funded. And what did they find? Improvement in facial skin hydration, elasticity, and roughness from collagen supplementation. This one, no, was not funded by industry, and this one showed to lead to more improvements in facial conditions. Okay, so that's 2 positive studies not funded by industry. This next one, again, no, improve human skin hydration, elasticity, and wrinkling. Okay, so that's 3. Then if we keep going down here, we've got some more. This one is also no, and it says demonstrates significant improvement in skin elasticity in sun-exposed areas. Okay, so that's 4. And we've got a 5th one here. So again, no, helps reduce water loss in middle-aged women. So that's 5. And then we go to this one, no, do not have benefits for forearm skin. So that's 1 out of 6 studies at this point that have indicated that there's no benefit of collagen supplementation. And finally, this last one is also no, can safely enhance human skin wrinkling, hydration, elasticity, and whitening properties. It's kind of written kind of funky, but they're actually saying the opposite, as in it's not increasing skin wrinkling, it's reducing skin wrinkling. So it's improving our skin as a whole. So what does that tell us? Well, it tells us that 6 out of 7 non-pharmaceutically funded, or I guess industry funded studies indicate that there's still a benefit of collagen supplementation. And that's where I want to go back to this term of significant. So when you're talking about, researchers use very dry language when they speak, and it's incredibly precise in that they're only talking about statistics. So statistically speaking, it is true that this analysis, when you do that subgroup analysis and break it off based off of the different funding sources, that the non-industry funded studies do not show a statistically significant effect of collagen supplementation. So he's absolutely right. The researchers aren't, duping us or anything like that. But clearly, as you saw, 6 out of 7 of the studies show that there's an improvement from collagen. So what that tells me is that A, either the effect is more mild, but there is still an effect, or B, you just don't have enough studies, because once you've eliminated a lot of the industry funded studies, now you're left with a smaller pool of data. And that means that you have less precision in the statistical analysis to actually be able to detect an effect. So if that's the case, then you wouldn't detect an effect, even though there might be one, just because the effect is more mild and because you don't have enough data to detect that effect. So that kind of leaves us in this gray area of, it's not that we shouldn't trust this analysis necessarily, but there's a lot more nuance to it than just reading the discussion saying, well, there's no significant effect. They didn't use the word statistically significant, but that's what they meant. There's no significant effect when you're looking at the non-industry funded studies. Another thing that he mentions is that there should be consistency between subgroup analyses. I definitely disagree on that front, simply because you can have a subgroup analysis that's based off of a different criterion. So you can look at like men versus women, and maybe for women you'll see a benefit, but for men you won't. And that may just be because of sex differences. That's a perfect example of where you might see differences in subgroup analyses, but that's a little bit of a side point, so I won't belabor that point. Okay, let's touch on one last part, and then we'll finish things off.
It has not been consistent for collagen. It is now being marketed as a fitness supplement, which I find hilarious, since collagen literally has the worst amino acid profile of any protein source on the market. It is absurdly low in essential amino acids, specifically the branched chain amino acid, and specifically leucine, which is the amino acid responsible for stimulating muscle protein synthesis.
Okay, so he touched on it there, and I agree with him completely. Collagen should not be marketed as a fitness supplement. It's absolutely atrocious as a protein source for any sort of muscle growth. And there have definitely been studies, and he talks about this as well, where if you compare collagen versus like whey protein or even other types of protein, and then look at the muscle protein synthetic nature of that molecule, that protein, collagen gets absolutely destroyed. So on that front, I completely agree with them. I do think it's laughable that it would be a supplement for fitness, unless maybe for joint health, which is a different discussion that maybe I can get into in the future. And I actually have an analysis on that as well, which is for the main channel. So maybe I'll link that in the description. So what do we take away from this? My takeaway from this is that this analysis actually isn't as much of a stake in the heart of vampiric collagen. And that's because I think that the effects are exactly what I've always kind of thought that they were either mild, or it's also still possible that there's no effect. And really the only reason why I'm saying that is, well, I guess two reasons. One, I would like to see more data specifically from non-industry funded studies. But the second reason is because we don't have comparisons against protein, or not enough comparisons against protein. So I think that would be a better comparison than just looking at placebo. I think reliably over and over again, even in the non-industry funded studies that we briefly touched on, it shows that collagen supplementation seems to improve indices of skin health and aging and whatnot when you're comparing against the placebo group. But we don't really have any comparisons against protein, and that's where I would really like to know if there are any actual differences. One last thing I'll quick mention, so why the collagen peptides was so important to point out, is that the belief is that the collagen peptides are bioactive, meaning that the cells are not just taking up the constitutive amino acids and then turning those into collagen. It's not just that mechanism. That is one of the mechanisms that's been proposed. But the more important mechanism, specifically to peptides, is that they can be absorbed, taken up inside the cell, and they can lead to changes in cell signaling that encourage the production of collagen. So they would actually try to stimulate more collagen deposition by the cells, which would not occur necessarily if you just douse the cells with the building blocks, the single amino acids that would be there for collagen synthesis. Okay, hopefully you found this informative. I think Dr. Layne Norton does great work. I'm not entirely convinced by the collagen literature myself, but I think that using this analysis as the end-all be-all is a mistake for some of the reasons that I mentioned, and some reasons I'll be getting into in my deeper analysis for the main channel. Anyway, thanks for stopping by. See ya.